Welcome children. Moving into the 8th lesson, Human Health and Diseases. Today's topic is immunity in video number 1. What is immunity? Immunity is the ability of the host to defend or fight against disease causing microorganisms. So what is immunity? It is the ability of our body or the ability of the host to fight against disease causing organisms. Immunity is classified broadly into two types, namely innate immunity and acquired immunity. First difference, innate immunity, it gets its name because it is inborn. By birth we have this immunity. We all are endowed with immunity right from birth. So innate immunity means it's inborn, immunity that is inborn from birth. Whereas acquired immunity means we acquire it during our lifetime. It is acquired during our lifetime either by contracting a disease or by a vaccination. So once again, the types of immunity gets its name from this point. Innate immunity is immunity that is inborn, whereas acquired immunity is the immunity that is acquired during the lifetime of an organism or of the human beings, either by contracting a disease or by vaccination. So it is acquired. Second, innate immunity is non-specific, whereas acquired immunity is pathogen specific. And the third point, innate immunity is accomplished by different barriers, namely physical barrier, physiological barrier, cellular barrier and cytokine barrier. So once again the third point, innate immunity is accomplished by 4 barriers, namely physical barrier, physiological barrier, cellular barrier and cytokine barrier. Whereas acquired immunity is accomplished by memory or characterized by memory. There are 2 types of response involved here, primary immune response and secondary immune response. This means that when our body encounters a pathogen for the first time, it produces an immune response which is of low intensity. And this response is called primary immune response. But when our body in the subsequent encounters with this particular pathogen, so in subsequent encounters with the same pathogen, our body elicits a very stronger or intensified immune response, and that is called secondary immune response, which is characterized by high intensity. Clear? So once again the meaning of memory or why we say acquired immunity is characterized by memory. This means that when our body encounters a pathogen for the first time, it produces an immune response, but that immune response is of low intensity. That immune response is known as primary immune response. But in subsequent encounters with the very same pathogen, our body produces an immune response of very high intensity or very strong intensity. That is known as secondary immune response. That's why usually certain disease like chicken pox, when you encounter it for the first time, this virus, you get the disease. But in subsequent encounter, the next year you are encountering a person having chicken pox, you do not get the disease. That is because your immune response is very strong, of high intensity, you do not succumb to the disease. Clear? You got it from this particular disease, chicken pox. For first encounter you get the disease, but in subsequent encounter with the very same virus that causes chicken pox, you do not get the disease because our immune system has produced a highly intensified immune response. So there are 2 types of immune response, very mild immune response which is called primary immune response, and a very strong or intensified immune response which is called secondary immune response. Okay, so this is the meaning of acquired immunity is accomplished or characterized by memory. Now moving into the details of the various barriers as I have told you under innate immunity. Physical barrier. Physical barrier, the main physical barriers are skin. It prevents the entry of microbes. So, the first line defense mechanism is our skin. The skin covering our body prevents the entry of microbes. If we didn't have the skin, or the skin, if there is a bruise or tear or cut which is open, we are prone to attack of microbes. But here the skin is covering our body, it prevents the entry of microorganisms. Not only skin, there are other secretions like the mucus. The mucus is lining our gastrointestinal tract, it is lining our respiratory tract, it is also lining our urogenital tract. It prevents the entry of microbes. And not only that, the ear canal is also lined by a secretion called earwax. Okay, that also prevents the entry of microbes. Physiological barrier involves secretions like acid from the stomach, tears from the eyes, and the saliva of the mouth. It kills the microbes that have entered our body. If it has entered our mouth, it kills by the saliva. Saliva is slightly acidic, or it has confronted this saliva and got into our stomach. There we have the acid secreted by the gastric glands which kills the microbes. Then the eye is cleansed, or the microbes of the eye are killed by the tears. Next we have the cellular barrier. The cellular barrier involves various cells like the natural killer cells, neutrophils, monocytes, and macrophages. These are various cells. The first three are circulating in our blood, whereas macrophage is present in the areolar tissue. How do they help? If the microbes have entered the blood or got into our tissues, what happens? They kill the microbe by a process called phagocytosis. Remember the process is phagocytosis. That is the way they defend. So, these are the different cells, the natural killer cells, neutrophils, monocytes, and macrophages. Macrophages is belonging to the areolar tissue. The remaining three are of the blood, circulating in our blood. They kill the microbes by phagocytosis. Now, the neutrophil, which neutrophil? It's PMNL. P stands for polymorphonuclear leukocyte. So, the which neutrophil is involved? It is PMNL. Last, we have the cytokine barrier. Cytokine barrier, it involves the interferons. These are proteins. These are proteins secreted by the viral infected cells. See, when our body is attacked by any virus, see, these are our cells, and it is attacked by a virus. These two cells are attacked by virus, which is indicated with red color. What happens is that these viral infected cells, so only these viral infected cells, I will indicate the cells that are affected by virus as yellow. These virus infected cells secretes a protein called interferons. Okay. So, they secrete a lot of interferons in its vicinity. And what is the role of this interferons is that it protects the unattacked cells from the attack of virus. So, it will not invade the other unattacked cells. So, once again, when a virus attacks our body cells, the invaded cells or the attacked viral cells of our body produce a substance called or a protein called interferons. And what happens is these interferons protect the unaffected cells from the viral infection. So, this virus will not attack the other cells, because already these cells which are attacked by the virus produces these chemicals called interferons. So, the other cells which are indicated in white are protected from viral infections. So, once again, a recap of the barriers. Physical barriers includes the skin, the mucus lining the gastrointestinal tract, the respiratory tract, and the urogenital tract. So, we know these tracts are lined by mucus. They prevent the entry of microbes. So, physical barrier prevents entry of microbes. Next, physiological barrier. These are secretions. The saliva of our mouth, the tear of the eye and the acid of the stomach. All these secretions kill the microbes that have entered. Then cellular barrier involves a number of cells like the natural killer cells, neutrophils, monocytes and macrophages. These kill the microbes by phagocytosis. And last we have the cytokine barrier. The cytokine barrier involves interferons. Interferons, what are these interferons? Interferons are proteins secreted by virus attacked body cells, and these interferons protect the unattacked cells from the attack of virus. So, this is a very good defense mechanism. So, once again a recap of the two types of immunity. First of all, what is immunity? The ability of our body to defend against diseases. There are two types of immunity, innate immunity and acquired immunity. Innate immunity is present right from birth, so they are called inborn immunity. Acquired immunity is acquired during the lifetime of a person, either by contracting a disease or by vaccination. Second point, innate immunity is non-specific, whereas acquired immunity is pathogen specific. Third point, innate immunity is accomplished by 4 barriers, namely physical barrier, physiological barrier, cellular barrier and cytokine barrier. Physical barrier involves skin, mucus lining the gastrointestinal tract, digestive tract, respiratory tract, etc. Prevents the entry of microbes. Physiological barrier, the secretions from the eye called the tear, secretion from the stomach called the acid, and secretion of the mouth called the saliva. All these kill the microbes that enter our body. Then we have the cellular barrier, which includes the natural killer cells, neutrophils, monocytes and macrophages. All these kill the microbes by a process called phagocytosis. And lastly, we have the cytokine barrier. Cytokine barrier are produced. They involve the interferons. These are proteins produced by viral cells, which protects the unattacked body cells from the attack of virus. Next, under acquired immunity, the third point we have is how is acquired immunity accomplished. Acquired immunity is accomplished or characterized by memory, and the memory involves primary immune response and secondary immune response. The meaning of this is when our body encounters a pathogen for the first time, it produces an immune response of low intensity, and this is called primary immune response. But on subsequent encounters with the very same pathogen, the body produces an immune response which is highly intensified, and this immune response is called secondary immune response. Hence we say our acquired immunity is characterized by memory. Now both these immune responses, primary immune response and secondary immune response, are mediated by 2 important cells, namely B lymphocytes and T lymphocytes. Both these lymphocytes are produced by the bone marrow, but some lymphocytes remain in the bone marrow and the others migrate to the thymus. You know, where is the thymus located? The thymus is located in the chest region, and it disintegrates as we move towards childhood, and late childhood it totally disappears. So it's in the thymus that some of the lymphocytes migrates, and hence it is called T lymphocytes. So both of them originate from the bone marrow, or they originate in the bone marrow, but after which some remains in the bone marrow and matures there. Some remain in the bone marrow where they mature, but the others they migrate to the thymus. You know, where is the thymus located? In the chest region. Okay, it is not present in you all because you all have covered your childhood. By 14 years it disappears. It goes to the thymus where it matures, and there it becomes the T lymphocytes. That's how we have the 2 lymphocytes, B lymphocytes and T lymphocytes. Once again I am reiterating, both the lymphocytes are secreted by the spongy tissue present in the long bones called bone marrow, but some remain in the bone marrow, some goes to the thymus and they mature. Those that mature in the bone marrow itself are called the B lymphocytes, and those that goes to the thymus and matures in the thymus are called the T lymphocytes. These two lymphocytes are involved in the immune responses, both the immune response, primary and secondary immune responses. Let us see the mode of action. Mode of action of the B and T. When an antigen enters our body, see I am going to indicate antigen red color because it is like an enemy. This antigen can be a pathogen, a microbe, it can be an allergen. We know a list of allergen, pollen grains, dust, etc. Or even it can be an insert. You know, under certain medical conditions, inserts are put into our body, maybe a pacemaker or stent, etc. So all these serves as antigen, foreign substance. When it enters our body, see, when it enters our body, what happens? The B cells are triggered. This triggers the B lymphocytes to secrete an army of chemicals. What are those chemicals called? Antibodies, or army of proteins called antibodies. And these antibodies fight with the antigen. There is a fight between the antigen and the antibody. So once again, the mode of action, when an antigen enters our body, maybe by an infection or by from the air or as an insert, whatever, when this foreign substance enters our body, what is triggered? The B lymphocytes are triggered, and the B lymphocytes are triggered, they secrete what? They secrete a number of antibodies, and these antibodies, like soldiers, they fight with the antigen. Clear? This is the mode of action of the B lymphocytes, whereas the mode of action of the T lymphocytes. The T lymphocytes by itself do not produce any antibodies. What it does is it helps the B to produce the antibodies. So, the T themselves do not produce any antibodies. No, they do not produce any antibodies by themselves. They help the B cells to produce antibodies. This is only during certain circumstances like organ transplant, where the T recognizes self from non-self, an organ, self organ from non-self, and then helps the B to produce antibodies. So remember, the T by themselves do not secrete antibodies. Instead, what it does, it helps the B. How it helps the B? It recognizes self from non-self, and then helps the B, and the B is only producing the antibodies, and these antibodies only fight with the antigen. Here, when is T coming to play is when there is a transplant. So, when an organ is transplanted. So, here the antigen is an organ that is transplanted. Clear? We will be coming to this once again. So, mode of action of B and T. B lymphocytes, when an antigen gets into our body, the B is triggered and releases an army of antibodies, and these antibodies fight with the antigen. But T does not by themselves produce antibodies, instead they help the B to produce antibodies, and T works only when there is an organ transplantation or a skin graft. Now let us see the structure of an antibody. So, I told you the B produces antibodies. So, let us see the structure of an antibody. The antibody consists of 4 peptide chains. Of these 4 peptide chains, 2 are heavy. 2 are heavy and 2 are light. So, the 2 heavy ones are this, which is running like a Y-shaped structure, and the two light ones are this. Okay. So that's all you need to know about an antibody structure. It has 4 peptide chains, 2 heavy and 2 light, and therefore it is referred to as, it also can be referred to as H2L2. Clear? But you have to learn the structure of the antibody, and remember, look at the ends of the antibody, look at these 2 arms, it is having a lock and key arrangement. Why lock and key arrangement? To show that antibodies are specific to antigens, not that all antibodies can fight any antigen, it is specific. Clear? Now there are different types of antibodies. Another name for antibodies, immunoglobulins. They are IgA, IgE, IgN, IgG and IgD. So there are 5 types of antibodies, namely IgA, IgE, IgM, IgG and IgD. Clear? So that's the structure of the antibodies, and I told you antibodies are the products of B lymphocytes. Only B lymphocytes can produce antibodies. Now based on the mode of action of the B and T lymphocytes, we are having 2 types of immunity: antibody mediated immunity and cell mediated immunity. Antibody mediated immunity is also known as humoral immunity because it is circulating in the blood. Okay, so there are 2 types of immunity based on the action of B and T, or which lymphocytes is involved. So remember, the first difference is what it involves. Antibody mediated immunity involves which lymphocyte? It involves B lymphocytes, and cell mediated immunity involves T lymphocytes. So the basic difference, or what difference differentiates AMI from CMI, is the type of lymphocyte that mediates it. So antibody mediated immunity is involving the B lymphocytes, and cell mediated immunity is involving the T lymphocytes. Then second point, how they act. As I told you earlier, how the B acts, once again I am reiterating. When an antigen gets into our body, the B is triggered to release what? Antibodies, and these antibodies fight with the antigen. Got it? I hope this flowchart will enable you to complete this. Once again, the mode of action. When an antigen gets into our body, and I told you the list of antigen, it can be different types of microbes ranging from bacteria, virus, fungus, etc. It can be an insert, or it can be a graft, or it can be an allergen. Okay, when any such foreign substance enters into our body, what happens? The B lymphocyte is triggered to release an army of soldiers. Here the soldiers are the antibodies, and these antibodies fight with the antigen. Okay. So this is the mode of action of the antibody mediated immunity. Now the cell mediated immunity. During organ transplant, see, when any organ is defective or non-functioning, we go for an organ transplant. It can be the transplantation of eye, it can be transplantation of the heart, liver, kidney, even it can be skin grafting from any part of our body. In all these scenarios, what happens is the T recognizes. Here is the role of the T. See, when an organ gets transplanted into our body, the T recognizes. So the role is the T recognizes the transplant. That means the T recognizes what? Self from non-self. Self from non-self. It differentiates self from non-self. And what happens? It helps B to produce antibodies. Now I am going to complete the same circle, and these antibodies fight with the particular transplant or organ. This results in rejection, organ rejection. That's why there is always rejection when we go for transplantation. Whichever organ or insert there is, to some extent our body rejects it, and that's why strong drugs are given, immunosuppressant, to suppress our immunity, to prevent this or to control this rejection. we are given immunosuppressants. So, once again here it is during an organ transplantation, what happens? The T lymphocytes comes into action. They recognize self from non-self, and they themselves do not produce antibodies. They help the B cells to produce antibodies, and these antibodies fight with the organ, and this results in rejection. Okay, and hence the person who has undergone an organ transplant is administered with immunosuppressants. So, once again, based on the mode of action of B and T lymphocytes, we have two types of immunity. They are antibody mediated immunity or humoral immunity and cell mediated immunity. The first point of difference, AMI involves the B lymphocytes and CMI involves the T lymphocytes. Clear? The second point is the mode of action. So, here you must specify the scenario during any infection. What happens? An antigen, when it gets into our body, what happens? The B is triggered to release an army of antibodies, and these antibodies, they fight with the antigen. Clear? Whereas in CMI, here you have to specify the scenario during an organ transplant or during an insert transplant or during a skin graft, whatever it be. When this particular foreign substance gets into our body, the B will not recognize, the T recognizes. The role of T comes here. The T recognizes self from non-self or differentiates self from non-self and helps the B lymphocyte to secrete antibodies, and this army of soldiers will invade and attack the organ that is being transplanted, resulting in rejection. Clear? So, I hope AMI or antibody mediated immunity and cell mediated immunity is clear, which involves the two lymphocytes, namely B lymphocytes and T lymphocytes. So, I hope today's topic you understood the types of immunity, which involves the innate immunity and acquired immunity, and the types based on the mode of action of B lymphocytes and T lymphocytes, AMI and CMI. Also with the structure, cartoon structure of the antibody. Thank you.